Good morning, distinguished guests, ladies and gentlemen. It is my great honor to welcome you all to this highly anticipated awareness seminar. Before we begin with the introductions of our special guests of honor, allow me to provide a brief overview of the initiative that brings us together today. Install Africa. Install Africa, the Innovation Science and Technology for Action Learning in Africa, is the result of collaborative efforts initiated in October 2023. This initiative leverages the Drosophila melanogaster fly model and state-of-the-art infrastructure to enhance fundamental research capacity in Rwanda. Rwanda has made remarkable strides in advancing its scientific capabilities, recognizing the pivotal role of research in sustainable development and public health. Install Africa was born from a collaboration between the Foresight Institute of Research and Translation, Rwanda, FIRAT Rwanda, and its technical partner, DROS Africa UK, in conjunction with key national institutions such as the National Council for Science and Technology, Rwanda Biomedical Center, the National Industrial Research and Development Agency, the University of Rwanda, and the Rwanda Food and Drug Authority. This collaboration embarks on a 5-year comprehensive capacity development plan aimed at cultivating the next generation of scientists and fostering research excellence across medical, applied, and fundamental sciences. From healthcare innovation to agricultural production, this partnership envisions driving progress in public health and sustainable development across Rwanda and beyond. It is against this background that we convene today, united in our shared commitment to advancing science, technology, and public health for the benefit of our communities. With that foundation laid, I am now delighted to introduce our special guests of honor, whose presence today underscores the importance of this endeavor. Today, we are privileged to have with us an exceptional panel of leaders, experts, and visionaries who have made remarkable contributions in their respective fields. Allow me to introduce our esteemed special guests of honor. First, we are honored to welcome Professor Abraham Mitike, the Principal of the College of Medicine and Health Sciences, University of Rwanda. Professor Mitike's leadership in medical education and research has been a beacon of progress in the healthcare sector. Next, we have Dr. Rujeni Mutimura, the Executive Secretary of the National Council for Science and Technology. Dr. Mutimura has been at the forefront of advancing science and technology policies that shape Rwanda's development. Joining us as well is Professor Claude Muvunyi, the Director General of Rwanda Biomedical Center. His dedication to strengthening public health systems has profoundly impacted the nation. We are also privileged to have Dr. Sekamo, the Director General of National Industrial Research and Development. Dr. Sekamo's work in fostering industrial innovation and development is truly commendable. Another distinguished guest is Professor Emile Bienvenu, the Director General of the Rwanda Food and Drug Agency. Professor Bienvenu's contributions to ensuring the safety and quality of food and drugs in Rwanda are invaluable. It is an honor to welcome Professor Leon Mutesa, a professor of human genetics at the College of Medicine and Health Sciences, University of Rwanda. Professor Leon Mutesa's pioneering research in genetics continues to break new ground in understanding human health. We also have Dr. Dieudonné Mutangana, the head of the Department of Biological Science at CST University of Rwanda. Dr. Mutangana's expertise in biological sciences has been instrumental in academic and research excellence. Finally, we are delighted to introduce Professor Janine Kondo, the CEO of the Center for Impact, Innovation, and Capacity Building for Health Information Systems and Nutrition, CIICHIN. Professor Kondo's leadership in health innovation and nutrition has made a lasting impact locally and globally. Please join me in giving a warm round of applause to our special guests of honor, whose presence graces this important seminar. Next, we will move on to narrating the profile of our invited guest speaker.
Ladies and gentlemen, it is my great honor to introduce an exceptional leader in science and academia, Professor Nadine Rujeni. Professor Nadine Rujeni is a leading female scientist and a trailblazing academic at the University of Rwanda, where she serves as a distinguished professor of infectious disease microbiology and immunology. Her career has been dedicated to driving health innovation in Rwanda and beyond, with a focus on the immunology, prevention, and control of infectious diseases, particularly malaria, neglected tropical diseases, and re-emerging viral infections. Trained at the University of Edinburgh, Professor Nadine Rujeni has held a series of progressive academic and leadership roles at the University of Rwanda, including Senior Lecturer, Dean of the School of Health Sciences, Associate Professor, and currently Professor of Microbiology and Parasitology. Her contributions span across teaching and curriculum development at undergraduate and postgraduate levels, student supervision, interdisciplinary research, mentorship of students and junior staff, and active engagement in community outreach and service. In addition, she has served on academic and management councils, chaired key school committees, and significantly contributed to staff capacity building and consultancy work. Professor Nadine Rujeni's research, supported by prestigious funders and published in esteemed journals such as Nature Communications, Science, PLOS Neglected Tropical Diseases, and BMC Public Health, has had a lasting impact on healthcare and disease management. Her recent studies include pioneering work on gut microbiota and malaria, the application of machine learning to the transmission of schistosomiasis, the investigation of cytokine kinetics in COVID-19, and public health research into HIV and neglected tropical diseases in Rwanda. Recognized nationally and internationally, Professor Nadine Rujeni is a Fellow of the Rwanda Academy of Science and an active member of the British Society of Parasitology and the American Society for Microbiology. She also serves as a reviewer for Springer Nature journals and has contributed her expertise as a doctoral committee member and a jury member for PhD evaluations at several international institutions. Her leadership extends to critical national health priorities, including membership in the National COVID-19 Task Force and service as a senior laboratory expert responding to Marburg and Mpox outbreaks. The career of Professor Nadine Rujeni is a testament to her commitment to advancing scientific knowledge, strengthening health systems, and mentoring the next generation of scientists. She is widely respected as a leading figure in infectious disease research and academic leadership across Africa. Ladies and gentlemen, please join me in welcoming Professor Nadine Rujeni. Professor Rujeni, the floor is yours.
Oh, okay. So I think now you can hear me. I hope. I finally managed to unmute. Can somebody speak and let me know that people are hearing me? Hello? Can I be heard?
Yes, ma, we can hear you, ma.
Okay, thank you very much. Good morning or good afternoon, depending on your geographical location, to everyone. It's a pleasure to be here with you today, and I'm glad to share with you some of the experiences that we've been through with regard to infectious diseases, immunomodulation. Um, I've kind of subdivided this talk into different sections, and I've put them here just to. Can I remove this private chat? Okay. Yeah. So briefly, I'll be going through what we mean by infectious diseases, and then talk briefly about the hygiene hypothesis, and then through immunomodulation, using some examples, some Helminth examples, malaria and COVID-19. And then later on, I'll explain a bit how we intend to leverage on the knowledge that we acquired through these infectious diseases, immunomodulation, to the gut microbiota, which is healthier than infecting people, and then some of the take-home messages and future directions from our group, but also inviting other researchers to try and continue asking the questions that are being raised by what we observe. Can you see my slide moving? So this is just a broad definition. If you Google it or ask ChatGPT or whatever, infectious diseases, what we mean by infectious diseases are those illnesses that are caused by pathogenic microorganisms. And these could be viruses, bacteria, parasites, or fungi. And so this whole group is what we call infectious diseases. And the particularity of infectious diseases is that they can spread directly or indirectly. What we mean by indirectly is that you may be infected by a disease from one individual, but the disease goes through a vector, through the soil, through water, so through another environment that is not the diseased patients. And so that's what makes it different from the non-infectious diseases or non-transmissible diseases that cannot be transmitted from person to person. So the WHO groups these diseases into three different categories based on how they infect people and the damage that they cause. So the first category is the one that cause high levels of mortality. So malaria, for example, is a disease that can cause high levels of mortality in endemic areas, or even people who are exposed as visitors. And then there are those diseases which place a heavy burden of disability in people. So the diseases that don't necessarily kill people, but leave them with certain levels of disabilities. And Helminth infections are an example of those. The third category is the category of diseases that I would put in um, so very fastly spreading, and that can cause global issues, global problems. So the pandemics, for example, the Marburg, COVID-19. So these are the kind of diseases that have global repercussions. So it's different from the two other categories, because the other two categories tend to be more endemic and localized in some areas and not necessarily moving around. So So when we talk about immunomodulation, the reason why we talk about immunomodulation, the whole story started with this guy, Stratton, in the 50s, when he was doing some epidemiological studies on children to try and understand what was causing eczema and asthma, and why some people were more prone to these diseases than others. And in his epidemiological observation, he noticed that children who were born from larger families and young children from larger families, so basically children who had many older siblings, they tend to be less susceptible to eczema and asthma, which are allergic diseases. And these allergic diseases, as you can know, they're associated with a poor level of immunomodulation. So he proposed that this resistance, if I can say that, to allergic diseases was because of the increased unhygienic practices from older siblings. So the most older siblings you have, the less likely you are to be hygienic. And so he was proposing that hygiene was at the like a factor for allergies. And so, following his observation, a lot of studies were looking at comparing the allergic diseases and autoimmune diseases from industrialized countries versus developing countries. And they were showing that in industrialized countries, you get more and more allergic diseases compared to developing countries. And they were concluding that it's because of the poor hygiene in developing countries compared to industrialized countries. And even some people, there were some anecdotal experiments where people could even infect themselves with Helminth infections or some kind of diseases to try and get rid of their asthma or asthmatic episodes. So at the immunological level, researchers were explaining this trend by the Th1, Th2 balance. So Th1, Th2 is T helper cells. So in immunology, in immune system, we have cells that are called T cells, and these T cells are in different categories. At that time, the category 1 and the category 2, or T helper 1 and T helper 2, were the ones that were very known. And scientists were explaining that if you have a high Th1 response, it will downregulate your Th2 response, which is associated with allergies. So the Th2 response is an immune response that is associated with allergies. I'll come back to this in more details. But then the Th1 is a more of an immune response that is there to deal with infections like viruses and bacteria. And so people with the bacteria infections and viral infections, they were proposing that they are less prone to allergies because they have a strong Th1 response. And so this was an explanation for several years. But then people realized that Helminth infections also induce a Th2 response. And yet people with Helminth infections were not prone to allergies. And like I said, in the anecdotal experiments, some people would infect themselves with Helminth and get rid of or reduce their asthma attacks. And so the problem again was, what about autoimmunity? Because autoimmunity also is associated with a strong Th1 response. So it was when in the 90s when Sekaguchi discovered T regulatory cells. So it's another component of the T cells that is more related to regulation, to immunomodulation, that people started to understand that so it's not just the balanced Th1, Th2, there is another component that parasites or pathogens are able to elicit or to induce that protects people against allergic diseases. So several years following the discovery of T regulatory cells, people started to understand the immune responses that are induced by Helminth, and later on, the discovery led to the fact that this immunomodulation or this regulatory response, this kind of responses that regulate the immune system against so to protect the pathogen or the parasite, was also strong enough to be expanded to bystander antigen, and this was what was protecting people infected with Helminth infection against allergic diseases. So it was in this period. So just briefly, an explanation of the immunomodulation. I apologize, this slide I thought was supposed to come earlier. So immunomodulation, the reason why it matters, why we are interested in immunomodulation. So as a definition, first of all, it's an alteration of the host immune response to foreign agents or pathogens. So the foreign agents don't have necessarily to be pathogenic. So the immune responses that we have could be against the harmless pathogens. So this kind of alteration is either through immune stimulation or immune suppression in order to maintain the desired level of host immune protection. So the purpose of immunomodulation is to kind of alter or change the immune responses against a foreign body, but to make sure that you maintain a certain level of homeostasis, if I can say that. And why it matters? For the urbanized societies, the immunomodulation matters because there's a rising incidence of chronic inflammatory disorders like allergies, autoimmunity, inflammatory bowel diseases. These are all chronic inflammatory diseases. And the interest is towards how we can induce immunomodulation and prevent or cure these chronic inflammatory diseases. And because pathogens have shown the capacity to induce this regulation or this immunomodulation, for the urbanized societies, it's very interesting because we could in the future be able to isolate the tools that induce immunomodulation and use them as treatment agents for people with chronic inflammatory diseases. In the developing world, we are burdened by infectious diseases that are killing or leading to disability. And the question is how we can induce this immunomodulation to prevent severe disease. So for us, for the developing world, it's more important to understand how diseases can be modulated so that the hosts are not killed by the pathogens, and also to try and understand what are the implications of this immunomodulation to vaccination, because when you are vaccinating people, you want to induce an immune response that is effective enough to the infectious diseases, but you need to be able, immunomodulation is a kind of suppression we've seen, so it's a kind of suppression of this protective immunity. And so when you're designing vaccines, you need to be able to understand what the role of immunomodulation and how they can affect vaccines. So Helminth infections. So we're starting with Helminth infections before we talk about Helminth-induced immunomodulation. I'll just take a few minutes to talk about the Helminth infections. I do apologize if I'm repeating things that some of the people here know. It's just because I didn't know the kind of audience that I would have. So I put this slide to just highlight the type of, so what is Helminth infections? They are due to Helminth parasites, and the Helminth parasites belong to two groups or two large groups in terms of systematics. So we have the nematodes or the roundworms for the layman, and we have the flatworms or the platyhelminths. So the roundworms, in this category, we have the soil-transmitted helminths and the filaria worms. And in the flatworms, we have the trematodes, for example, Schistosomiasis, And we also have the tapeworms or the cestodes, and in this category, for example, we have tenia. They were common in most parts of the world 60 years ago. So these are parasites that humankind has evolved with, but at the moment they are more prevalent in sub-Saharan Africa, in Asia, and South America, whereas in the Western world you see less and less Helminth infections. They're responsible for considerable disabilities, including blindness and elephantiasis, or the filaria worms. Those in West Africa, that they are responsible for things like blindness and elephantiasis. They're responsible for anemia, stunted growth, poor cognitive development, and malnutrition, especially in children. And we've seen, for example, some programs in endemic countries, so countries where Helminth infections are present. They tend to have control programs, even here in Rwanda. For those who are from Rwanda, you know that we have programs to deworm, to treat children who are infected with Helminth, to try and reduce these kind of disabilities and improve cognitive development of these children. They also have a negative socioeconomic impact in poorest communities, because if you have these disabilities, if you're blind or you have elephantiasis is this disability where you have very swollen legs, and it's very difficult to work, and it's difficult to do your everyday work. So productivity reduces, and this has a huge socio-economic impact. But they also have a burden on the health systems, because we have to deal with this. So we have to invest in deworming, you have to invest in prevention and all that. So Helminth infections, basically, they are negative for us. So what is Helminth induced immunomodulation? In general, the Helminth infections, like I said, they don't necessarily lead to high mortality. So they tend to lead to disability, but they could also stay in the host without causing any signs and symptoms. And they can establish chronic infections, because they can survive in the host for several years, and sometimes they can they can stay for an entire lifetime of the host. So in vitro, it's been demonstrated that the immune system is capable of parasite expulsion. So it's capable of eliminating parasites. But in animal models, this has been shown to be accompanied by tissue damage. But in natural infections, the tendency is that these parasitic infections, they tend to mute the immune response. They tend to reduce the kind of immune responses that the host mounts, and they tend to be accommodated by the host. And so the host tends to tolerate the parasites. And this has applications both for the host and for the parasites. So for the hosts that are able to tolerate these parasites in endemic areas, they tend to have less physical damage. And for the parasites, of course, this tolerance by the host will allow the parasites to survive for a very long time. So Helminth infections are accompanied by a strong regulatory responses, and these regulatory responses are the ones that allow them to survive in the host. So immunologically, the immune responses or protective immunity against Helminth is characterized by a Th2 response, like I said earlier. And but in addition to the Th2 response, they also have a regulatory response. So these strong regulatory response restrict pathology in the hosts, and studies have shown that these regulatory responses span from the innate immunity all the way to acquired immunity. So there are molecules that are involved in immunomodulation, all the way from the toll-like receptor signaling. So where in innate immunity we have receptors that sense the parasites, sense a foreign body. These toll-like receptors are like the door to the innate immunity. And so at this stage already, when you have a Helminth infection, there are molecules that were identified to be involved in immune regulation. So Helminth infections are really strongly modulated. And this regulatory network that is induced by Helminth infection is also thought to temper responses to non-Helminth antigens. So like I said, all the way from the hygiene hypothesis, scientists have observed that this regulatory network that is induced by the Helminth infection are also able to temper with allergens and really reduce or shut down allergic episodes. When I started my PhD a few years ago, the mechanisms behind the modulation or the regulation of immune responses towards allergens to prevent or alleviate allergic diseases was not very well understood or very well explained. And this is how we kind of tried to understand the similarities and the differences between the Th2 response that is induced by Helminth infection and the Th2 response that is induced in allergic diseases. On this slide, I don't know if I have a pointer, but on the left-hand side of my slide, you can see immune responses in Helminth infection, and there's a two stage. So the primary infection, we mean primary infection by infection with when a host is exposed to Helminth infection for the first time. So this is the kind of response that you get, and this was a desk review comparing the primary infection with the allergic responses. So you can see that all the components that you see in allergic responses, so you can see the dendritic cells from the epithelial cells, so the dendritic cells presenting antigen to Th2 responses, and the Th2 cytokines that are elicited or produced that activate effector cells, so eosinophils, mast cells, B cells, basophils. And these effector cells, this activation of the B cells in this case leads to strong IgE responses, so antibody responses of the IgE isotype, which is characteristic of the allergic diseases. So you can see if you compare this response to the primary response, it's pretty much the same. So you see effector responses, and you see B cells that are activated and that are producing IgE responses. So it was very interesting for us to see the similarities in terms of the IgE responses. But what was interesting was that when in secondary or chronic infections, which is very frequent in endemic areas where you have heavy infections or chronic infections, people who have been exposed to these Helminths for several years, we could see that so instead of just limiting the response to the Th2 response, there was another response, a regulatory response, so T regulatory responses and B regulatory responses that are associated with cytokines like the TGF-beta, the IL-10, and the CD23 in terms of B cell responses. This CD23 is a low affinity receptor for the immunoglobulins, for the IgE. So for the chronic infection, in addition to the IgE, you could see a strong IgG4 response. This immunoglobulin is different from the IgE response. They tend to have the same target, the same receptors, but the IgE response is more of a blocking antibody to the IgE. So people with chronic infection tended to have more IgG4 responses compared to IgE responses. So we did look at people who were exposed to schistosomiasis. This was a study in Zimbabwe. We looked at people who were exposed to schistosomiasis, which is one of the Helminth infections, and we tried to understand their regulation of the IgE and the IgG responses, and we tried to look at whether these immune responses, these antibody responses, were related to their allergic responses. So this is an illustration of a study that was looking at the allergic responses from people in endemic areas. So the SPT means the skin prick test. So we were doing a skin prick test to check whether people are allergic. And on this graph, you can see that those with high IgE responses, antigen-specific IgE responses, and total IgE response, so non-specific and antigen-specific responses, had higher levels of skin allergies. And what was interesting for us was this negative correlation between the skin allergy and the infection with schistosomiasis. So people who had a strong or a heavy infection with schistosomiasis tended to have a lower risk of atopic responses or allergic responses on the skin. So this was very interesting for us, and we really wanted to explore this further, because at that time there were some studies in allergic patients that were showing that the CD23, like I said, CD23 is a low affinity receptor for the IgE. So IgE has a high affinity, the FcR1, which is a high affinity receptor, but it also has a low affinity receptor. And those of you who are familiar with immunology, you know that for an immune response, for an antibody response to be effective, you need to have a strong binding. So you have to have a high affinity. So the CD23 has a low affinity with IgE, and in allergic patients, studies were showing that those with a strong soluble CD23 response, they tend to have less allergic episodes, and we wanted to see whether this CD23 was involved in the regulation of the IgE in endemic areas in Zimbabwe. And this is what we observed. So we were looking at two different villages with differing infection levels, because at that time the animal models, the studies in mice were showing that mice with heavy infection tended to have less or no allergic diseases compared. So if you have a mice with heavy infection, you could induce allergic responses, and they were not susceptible. So we had to do a comparative study where we compared low infection village with a high infection village, and we're looking at the level of CD23 that they had, and we could see that in high infection area, you had more CD23 responses in their blood, and this CD23 response was associated with negative skin allergies, and in addition, it was also negatively associated with infection intensity. So basically, you could see that the CD23 was involved in the regulation of the IgE response, the antigen-specific IgE response, and the antigen being the antigen from the parasite, but also antigen from the allergen. So here down, it's a negative correlation between the DERPI. So when we were doing the skin prick tests, the house dust mite was the allergen that was showing a higher prevalence. So we tested with antibodies, anti-mite antibodies, to see whether they were correlated also with the CD23. So at that time, really what we were putting forward was that the CD23 was something that could be involved really in the regulation of the allergies, because it's a low affinity receptor with the IgE, and it tends to activate more of the B cell responses, which is more of the B regulatory responses, rather than the mast cells, which are involved in histamine release and allergic responses or allergic diseases. So, following my PhD studies, when I came back in the country, I was involved in trying to understand malaria, because malaria at that time was even a bigger issue than schistosomiasis, because as you know, malaria has a higher mortality rate, and it affects around 200 million in the world. Even up to now, it kills several thousands of people every year. But what was interesting for us at that moment, following the exposure to the immunomodulation with parasites, was to try and understand why do some people have severe malaria, some others have a mild malaria, and some people even live without ever getting malaria, even if they're exposed to the parasites. And what we thought was that the immunomodulation was involved in the clinical presentation of malaria. And around the 2017 and forward, we were privileged to have funding to try and understand. But before I dig into our studies in malaria immunomodulation, allow me to take you through these immune responses or protective immunity in Plasmodium or in malaria. So as you know, malaria is due to a parasite that is called a Plasmodium. We have several species, and these parasites, we get them through mosquitoes. So they are transmitted by mosquitoes. And you can even see here, this is an illustration of a mosquito. But why I'm bringing this life cycle of Plasmodium is to show you that at different levels, we have different kind of immune responses, protective immunity, inflammatory responses against the parasites. And we have different types because even within the host, the Plasmodium has different shapes. So it has different stages, and you could call them different shapes. So when you get bitten by a mosquito that carries Plasmodium parasites, at this stage they are called sporozoites. So you get a different type of parasite from, for example, the one that you find in the liver. So the parasite that gets through the skin has an opportunity to travel and go through the blood vessel all the way to the liver. But in this journey, even if it's a very short journey, the parasite primes an immune response. It provokes an immune response from the host. And of course, at this level, the parasite is highly antigenic. And I'm not sure I mentioned this, but parasites, in addition to inducing an immunomodulation, they also try to change their shapes several times to make sure that they survive in the host and they are not recognized by the host immune responses. So the parasite moves all the way to the liver, and in the liver, they are protected by the fact that they're able to penetrate the liver cells, the hepatocytes. So the parasite penetrates the liver cells and survives inside the liver cells. And you can see that, for example, innate immunity is kind of blocked. It cannot reach the parasite. And inside the liver cells, that's where the parasite flourishes and multiplies and release another stage of the parasites that we call the morozoites. And these are the ones that go into the blood vessels and now start to infect the red blood cells. And this is when we start observing clinical symptoms for those who are susceptible to clinical signs. So generally, as a basic kind of immunology, we get responses. So once a red blood cell is infected, there are some antibodies that can recognize, that can bind to it and induce or attract phagocytes that are going to be able to eliminate the infected red blood cells. But again, at this stage, the parasite has an extremely high antigenic variability, because this is an area where the parasite is exposed to a strong immune pressure. It's exposed to several immune responses, so the innate macrophages, the natural killer cells, the antibodies. So this is where the parasite really has to be highly antigenic to be able to survive. And in addition, also it should be able to induce regulatory responses that are going to shut down or suppress these effector responses. So in the gut, in the mosquito, you also have different stages that also are exposing the parasite to immune pressure, but the immune pressure is very limited, and the antigenic variability is low in the mosquito. So from the observation from the literature, it was apparent that parasites, so Plasmodium and the hosts, interact with the complex cellular immunity, and we could see that T regulatory cells, or TGF beta, were involved in lots of experimental models that were showing that mice with strong regulatory T cells and TGF beta were showing a lower susceptibility to severe malaria. And so we wanted to understand how these regulatory cells are regulated, basically. And like I said, in the 2017, we managed to get a grant to try and understand how immunity is regulated. This cellular immunity is regulated in patients who are exposed to malaria. And so we were looking at different populations, so people with severe anemic malaria and people with mild malaria, to try and understand how their cellular immune responses were regulated. And of course, because we could not dissect these people, what we did was looking at the cytokines, and cytokines are kind of molecules that are involved in regulating different type of cells. So we were looking at cytokines that are involved in, for example, activating regulatory T cells, in cytokines that are involved in activating Th1 responses, cytokines that are involved in activating Th2 responses. And the reason why we were looking at these cytokines is because looking at cytokines can give you an indication. It's an easy approach to understand the mechanisms of regulation without invading the host, just trying to understand the systemic regulation of these cytokines. And so from the exploration of these cytokines, the outcome can allow you to dig deeper, but knowing what mechanisms, what cells are of interest. And of course, in addition to the cytokines, we were looking at the transcription factors for the T regulatory cells. So FOXP3 is a transcription factor. It's like a gene that leads to the regulation of the T regulatory cells, or to the production of the T regulatory cells. So on this slide, it's just summarized. It's a study by Ella Raisa, who was a PhD student in our group at that time. It was showing that if you look at TGF beta, which is a kind of regulatory cytokine, a cytokine that is involved in the regulation of the T regulatory cells, if you compare the levels of these cytokines, being TGF beta 1 and 3, in patients with severe malaria and those with mild malaria, you could see a significantly a strong difference between the mild cases and the severe cases. So you can see a very high level of TGF beta compared to the TGF beta in severe malaria patients. And IL-9 is also another regulatory cytokine, another cytokine that is involved in the regulation, in the regulatory network of parasitic infections. And it has also been shown to be involved in the regulation in chronic inflammatory disorders. And you could also see that the IL-9 was really upregulated in mild malaria. So patients with mild malaria had a tendency to have a higher levels of these cytokines that are involved in the regulatory network of malaria. And as a confirmation, if you look at the RNA expression of the FOXP3, which is a transcription factor for the T regulatory cells, you can see that you have a higher expression, stronger expression of these FOXP3 in mild patients, so patients with a mild disease, who just had a very really mild disease. They had malaria, comparable parasitemia, but then they had fever, headaches, and that's it. They had medication, and that was it. But those with the severe malaria had anemia, seizure, and all that. So, in addition to these TGF beta and IL-9, we could also see that people with severe disease, severe malaria, so this is an analysis that groups people with the same kind of levels of cytokines. It's a principal component analysis. So you have a grouping of the people with the same levels of cytokines, and you can see that this blue color is a group of those people with the severe disease, and the red color is people with the mild disease, and then you can see the negative controls down here where you don't have any cytokine. Really, the cytokine levels were very low. So you could see that the people with severe disease, they have particular cytokines that they tend to have in upregulation. So you have they have higher levels of cytokines that are upregulated. But if you look at them in detail, it's more the Th1 response, for example, the interferon gamma is a Th1 cytokine. You have it higher in severe diseased patients, but you have a lower levels in those with mild disease. You can see what was interesting. So the IL-10, IL-17, IL-7, they are more involved in the regulatory network. So it was expected to see them down-regulated in those people with severe malaria. But what was interesting was that the theory of the Th1 and Th2 balance that I started off with was a bit challenged here, because you can see a Th1, the Th2 cytokine and the Th2 cytokine here that are pretty much upregulated as well, in addition to the Th1 response. So people with severe disease really they tend to have what scientists recently called the cytokine storm. So they tend to have an upregulation of several cytokines, but most likely more the ones that are prone to inflammation, that are leading to inflammation, and this is why they have a severe disease. So this study occurred a few months, if I can say that, prior to the COVID-19, and we wanted to check whether the patterns that we're observing in malaria, which is a parasitic infection, could also explain the disease severity in COVID-19. So we did a longitudinal study looking at COVID-19 patients, looking at the same cytokines and also the transcription factors to try and understand if we could see the same patterns. Interestingly, for the COVID-19 patients, those with severe disease really tended to have almost all the cytokines upregulated. So as opposed to the malaria patient who had mostly the pro-inflammatory cytokines that were upregulated compared to the regulatory cytokines. For the COVID-19 patients, those with a severe disease really tended to have a strong upregulation of pretty much all the cytokines. And this is again a study by Dorothy Mai, who was doing her PhD as part of the project. And what was interesting was that because this was a longitudinal study, and the reason why we did a longitudinal study is because we wanted, this was kind of a new disease, that the immunogenicity of this virus was not really very well understood, and we wanted to understand the kinetics, the evolution of these cytokines in those people who end up recovering or end up dying or end up not really developing a severe disease at all. And what was interesting was that the TGF beta, so the regulatory responses, so the TGF beta, the 1, TGF beta 3, and IL-9, even if they were highly upregulated in the patients with a severe disease, they tended to stabilize and be increased mostly in those with a mild disease. So the tendency that we were observing in malaria patients Can you hear me now? Okay, I think you. Can you hear me now? I think it's back. I had some network issues. Can you hear me now? Well, at least I can move the slide and I can see some green light. It's no longer red. Okay, good. So what I was saying was that the observation, the upregulation, the regulations of the cytokines in COVID-19 patients, when we look at people with the severe disease and compare those who ended up dying and those who ended up recovering, we could see that there was a strong, a significant difference between the two groups in terms of the regulatory, so the interferon, the interleukin 9, so the IL-9 was really upregulated in those patients who ended up recovering versus those who ended up dying. And what was interesting was that the pro-inflammatory cytokines, the Th1 cytokines, were upregulated in those people who ended up dying compared to those who ended up recovering. And if you do a ratio between the two, you have a significant difference between the two. So basically, this was showing us that in those people with the severe disease, even if they have an upregulation of all of the cytokines, they don't tend to have high levels of IL-9, and the IL-9 level has a tendency to regulate the other pro-inflammatory cytokine. So that was the conclusion of the study by Ella Larissa. And so from the observations, the epidemiological studies in Helminth, malaria, and COVID-19, we've seen that immunomodulation is something real, but the question still remains on how we can explore the immunomodulation. To going back to the initial questions that I highlighted, to kind of try and answer the questions of how can we prevent or cure chronic disorders, or how can we modulate our immune responses to pathogens to make sure that we are leading, or we are inducing a mild disease to try and alleviate our health system as well. So there is a hypothesis that evolved from the hygiene hypothesis. So like I started off explaining the hygiene hypothesis, there has been an evolution in the hypothesis, and currently the one that is more realistic is the old friend hypothesis. This is a hypothesis that states that the raising failure of immunoregulation is due to inadequate exposures to microorganisms. And these microorganisms are not necessarily the pathogenic ones. The microorganisms that we are talking about here include the microbiome. So the commensals to which we are exposed to that can drive the development of the immune system and drive the expansion of these regulatory cells that are of interest in terms of controlling the chronic diseases, but also infectious diseases. So many of these organisms are symbiotic, like I said, and they are components of a healthy microbiota. So it could be the gut microbiota, it could be the skin microbiota, it could be the lung microbiota. So we have a huge population of microbes that live in our body, in different parts of our body, that are able to elicit an immune response and that are able to modulate the immune response the same way that we've observed in infectious diseases. And what we wanted to understand, like I said, in the developing world, what we are interested in is what can help us modulate or reduce or minimize the severity of the infectious diseases to which we are exposed to. So trying to understand the modulation or the immunomodulation or the modulation by the microbiota by Mutoni as students, PhD students in our group. Again, connection is lost. Okay, so it's not going. So Mutoni started off his PhD trying to understand how the gut microbiota could be associated with the clinical presentation of malaria, because it was the same project of trying to understand the regulation of severe malaria. And so he tried to understand. So this is a study that he published trying to show that the gut microbiota is involved in the modulation of the immune system towards malaria, and that for example, some studies were showing some antibodies that are elicited by the gut microbiota, the anti-alpha-gal antibodies that are involved in reducing the transmission of malaria, or reducing the interfering with the migration of the plasmodium from the skin to the liver. So the alpha-gal antibodies are kind of protective antibodies in malaria, and studies had shown that the gut microbiota can elicit these antibodies. So it was very interesting to see whether, in addition to inducing protective immune responses, it could also be involved in the severity of the disease. So what he did was to look at 3 groups of malaria patients: those with severe malaria, those with asymptomatic malaria, and those with mild malaria, and look at their stool samples and analyze the gut microbiome, so the bacteria in their gut, looking at the 16S sequencing to see the differences and whether there was an association between the composition of the gut microbiota and the clinical presentation of malaria. So briefly, I'm conscious of time. So what he was able to show, this is a manuscript under preparation, what he was able to show was that people with severe malaria and those with mild malaria and asymptomatic malaria, they have different composition of their gut microbiota. So the microbes or the bacteria composition, so the diversity in their gut, in their intestine, in terms of the bacterial population, is different. Those patients with severe malaria could cluster. So this is again a principal component analysis. You can see that the people with the severe malaria really cluster separately from the rest of the group. And you can see another group down here, the blue one, the asymptomatic patients. You can see really separation between the two groups. So big differences between the two groups. And if you look at this figure here, what that is showing the alpha diversity between the groups, you can see a significant difference between patients with severe malaria and those with mild malaria, and a significant difference between severe malaria and those with a symptomatic infection. And so this was really a confirmation that the gut microbiota could be the one that could be explored as another avenue to explore the immunomodulation without having to infecting people with pathogens. The take homes from what I've been talking about over the last almost 1 hour is that infectious diseases can induce regulatory responses from the host. And this regulatory response, what we call immunomodulation, allows their survival in the host, but also protect the host against excessive inflammation or severe disease. But also this kind of immunomodulation can extend to bystander antigens like allergens and other inflammatory agents, and protect the host against chronic inflammatory diseases. Commensal microorganisms could induce immunomodulation the same way as infectious diseases, and an understanding of how they are regulated and how they influence this immunomodulation will help us understand or lead to practical applications in terms of, for example, can we change, because the gut microbiota, for example, is influenced by the nutrition. So one of the application could be, for example, changing the nutritional habits and therefore changing the microbiota composition and changing or inducing or modulating the immune responses that is against the infectious diseases, but also chronic inflammatory disorders. So what's next? At least in our group, we are trying to correlate immune responses. So the study by Mutoni, we are going to complement it with looking at immune responses and looking at how they correlate with the gut microbiota diversity. And later, hopefully understand the mechanistic interactions between the two. So trying to understand the functionality of this gut microbiota, what genes, for example, are involved in the regulation of the immune responses and stuff like that. And for example, how nutrition can influence the gut microbiota. There's another PhD student who's looking at the nutrition aspects of so how the nutrition is influencing the clinical presentation of malaria, and we could link and see what how this network is all regulated. And some of the applications, like I said, could be changing the nutrition or identify disease markers, so severe disease markers. So if we could early on detect markers of severe disease, so people before they develop a severe disease, if we could get to know what immune markers can be used as a proxy to make sure that we do better management, and also some markers for immune protection. So all of these, so immune protection for vaccine development. So when you're designing vaccines, we know, for example, for malaria, for schistosomiasis, we haven't, well, for malaria, there are some vaccines, but their efficacy is still low and their availability is still very low. And so the march towards development of vaccine is still very open, and understanding this immunomodulation and this immune protection could be an avenue of research. So thank you very much for your kind attention, and I'd like to thank our current PhD student and former PhD student and collaborators from the Université Catholique de Louvain, from the University of Edinburgh, and from the University of Rwanda. Thank you very much, and sorry for going beyond the allocated time, but I'm very happy to get questions. If you can give me 1 minute so that I can plug in my computer because I'm running out of battery, that'll be very helpful. Is it all together? Okay, yeah, I'm all set. Questions, comments,
Thank you very much,
interests?
Professor. Can you hear me?
If you can increase your, okay, or maybe okay, go ahead.
Can you hear me now, Prof?
Yes.
Thank you very much for this insightful presentation. I must say that you've really done a lot, and then we have learned a lot from your presentation. Um, Shortly, let me just like a brief recap without wasting time. I wouldn't want to give another a lecture to whatever you have said, Ma. Uh, Prof talked about the title of Prof's presentation was Insight into Infectious Disease Immunomodulation. She did talk about the basics of um, immune system, and then she moved into the primary infection, secondary infection, research work with schistosomiasis induced immunomodulation, malaria and immunomodulation. She talked about the fact that antibodies, uh, um, the antibodies response when infection is in the body, and that the antibodies elicit phagocytic cell. She talked about those primary mechanism of malaria movement in the body. She moved to the longitudinal research done with COVID patients, because the research was done very close to COVID period. Then she talked about the gut microbiota and how they also induce parasite immunomodulation, and the take home. I believe that this is the meat of what she presented. The fact that immune infectious agent can also induce regulatory response that can make the host vulnerable in such a way that they can also protect the host against excessive inflammation. And then she told us what next that she's planning to do in her lab with her students. They are trying to look at the correlation between immune response and then gut microbiota, and also markers for, just to mention few of what Prof talked about. She talked about the fact that they are in their future studies, they are looking at the markers that can be used for vaccine development and design. Thank you very much. Now it is time for questions. So I'm going to look at what people are asking, Prof. Then I will read to you for your prompt response. I saw
There's
one
already an
question.
interesting one thing here. I was laughing at the thought because are there potential risks in eliminating helminth infections too aggressively without considering their role in immune regulation.
Okay.
Do you hear that? Yes.
I think.
So,
Please go ahead. Go ahead,
sorry?
Prof.
Yes. Like I explained earlier, a long time ago, people were interested and they could say that helminth infections are beneficial, and so the elimination of this helminth in the Western world is the one that led to the rise in allergic diseases or autoimmune diseases. But what I can say is that if you look at the burden of helminth infections, especially in young children, already that burden is really huge, and I wouldn't advise not to eliminate them because of that burden. But secondly, there are alternatives. So, like I said, immunomodulation or immune regulation is not done by just the helminth. We could rather explore, for example, the commensals, the microbiota, the commensal bacteria that can induce the same level of immunomodulation, could potentially induce the same level of immunomodulation by as the one induced by the helminth, and explore that one. Another possibility would be to rather isolate the molecules. Some groups have done that. For example, the groups of Rick Meisels, they have tried to isolate the molecules in the helminth parasites that can induce immunomodulation. So instead of infecting people with helminth or letting people infected with helminth, which is unethical because of the burden to the host, isolate the molecules that are involved in immunomodulation and use them in the treatment of chronic inflammatory diseases. But you also have the avenue of looking at the commensals. So to me, helminth infections should be eliminated because they cause a lot of damage in young children, and they're a burden even to our health system. Disease immunoglobulin vaccine response, yes. So there's someone who asked whether this immunomodulation affects vaccine responses. Does diet have any role to play in these? Yes, like I said, unfortunately, I didn't have time to go through some studies that have shown that helminth infection affects the vaccine responses negatively, actually. There's a recent study in Uganda that showed that people who have helminth infections and are vaccinated with the likes of BCG, for example, we give BCG to all children born in most of African countries. And so the studies highlighted that efficacy of the vaccine for those people who had helminth infection was really less compared to efficacy in people without helminth. And when you treated these children following or before, no, actually before vaccination, if you treated people before vaccination and you vaccinated them, the efficacy of the vaccine was improved. And so you can see that this immunomodulation, of course, is good when you're looking at the side of severe disease or severe pathological diseases. But again, most of these parasitic infections, for example, the helminth infection, they don't tend to lead to severe disease anyway. And so people who would benefit would be the people who are not exposed to this helminth. And so, does diet have any role to play in these? So diet, what we've observed is that it could influence the gut microbiota composition. And if you remember when what I said from Jean Damour's study, it's that people with severe malaria have a different gut microbiota composition compared to those with asymptomatic malaria, for example. So definitely the diet could influence the gut microbiota and influence the immunomodulation indirectly, because the gut microbiota is the one that influences the immunomodulation. How does malaria-induced immunomodulation influence hosts' susceptibility to co-infections or response to unrelated vaccines? Malaria-induced immunomodulation influence host susceptibility. Oh, yes. So there are several studies that have shown that co-infection, for example, malaria and Helminth. For example, there were studies in Senegal and a number of other countries that were showing that people with Helminth infection, for example, Schistosomiasis or soil transmitted, especially schisto, probably because it's a blood-dwelling parasite, the people who are exposed to schisto, some studies were showing that their clinical presentation of malaria was less severe. But there are some few studies that were also showing the opposite. So really the epidemiological studies comparing people with or without the other infections are showing some interactions, but of course the conclusion is not like clear cut, because in some population you can see some protection, in some other population you can see some susceptibility, so they become more susceptible to the other infection if they have Helminth infection, for example. But in some other studies, they were showing that if you have a Helminth infection, you are less susceptible to severe malaria, etc., etc. And I think the reason why we have this heterogeneity is because there are so many parameters involved. So different Helminth species lead to different regulation of the immune system. Different malaria species lead to different regulation of the immune responses, but also the hosts. We have a huge diversity in terms of how we respond to malaria, how to Plasmodium, how we respond to Helminth infection, and all these differences are really difficult to streamline and come up with a conclusion that, oh yes, people should have Helminth infection so that they don't have severe malaria, for example. That's something that you can never read in the literature. But in terms of vaccines, all of these parasitic infections will influence the vaccine responses because they all induce a regulatory response, and this regulatory network goes beyond the parasite to which that elicits them. So, for example, if you have malaria, sorry, you have a regulatory response that not only is effective against Plasmodium, but also can expand to other antigens. Is there a role of probiotics used among children in reduction of parasitemia? I'm not very sure there are probiotics that have already been developed to help reduce parasitemia, because I think the gut microbiota studies are still at early stages, and people are still trying to understand the mechanistics of these microbiota. And I haven't seen any study that is showing that this direct link between the probiotics and parasitemia. Can you elaborate on how the immune regulatory mechanisms differ between Helminth? Well, there are some differences. Of course, there are some differences even between different populations, but the key thing or the basics are the same, because In malaria, we see regulatory T cells, for example. We see regulatory B cells. We see the TGF beta. We see IL-9. The same molecules we see, and cells, we see them in Helminth infections. So the mechanisms of how all these cells work together to regulate could be different and different in different populations, but the fundamentals are the same. So the Tregs, if you isolate your Treg from a malaria patient and you isolate your Treg from schisto patient, you have the same marker of a T cell, of a Treg, but the proportions or the number of cells, the number of T regulatory cells, for example, they can differ. For example, we've seen that in Helminth, schisto-infected people who live in endemic areas, because these Helminth infections, unlike malaria, they tend to be chronic. So people with Helminth infection tend to have this infection for a very long time, compared to malaria, where when someone has malaria, they are treated and they get rid of the infection. So the regulatory responses, the regulatory cells, the Tregs, are likely to be less in terms of the population and the efficiency compared to the regulatory cells in the Helminth infections. But again, if you look at, for example, malaria patients with asymptomatic disease, because them also they have malaria for a very long time, so the quantity or the magnitude of the T regulatory cells could be comparable to the ones that we see in Helminth infections. But all of these are things to explore and see how best to use them to our advantage.
Thank you very much, Prof. Briefly, let me just ask that: is there any? Do you envisage any hyperimmune response to Plasmodium when patients are in the case of co-infection now with Helminth? Could there be any effect of anti-Helminth treatment, leading to a hyperimmune response to Plasmodium?
Oh, you mean if people are treated with
Yes,
anti-helminthics, if
yes,
their immune responses to Plasmodium is altered?
yes.
Yeah, of course. Like I said, there are several studies that have shown that this immunomodulation disappears when you treat people. There's even, I think we did one of them for schisto. So when you treat people, this immunomodulation, these immune responses that are associated with regulation, they tend to go down and even disappear. The trick is to make sure that you keep treating these people so that this immunomodulation is completely gone and they're not interfering. It's not interfering with the effector or protective immunity to Plasmodium. But again, protective immunity to Plasmodium, we've also seen that it involves also regulation, because effector responses or inflammatory responses are good at maybe they could be good at eliminating the parasite, but also damaging the host. So you need a balance between an inflammatory response and the regulatory response. But all I can say is that if the regulatory response is due to Helminth infection, once you treat the Helminth infection, the regulatory response goes with it.
Thank you very much, Prof. I must say a very big thank to the organizers as well for putting this thing together. I think that's the last question, Prof. Um, I believe that we must have had
Why
or
is it
learned.
that there's a question that is just popped?
Okay,
The
okay, go
patient
ahead.
will have all the clinical symptoms of malaria and yet test negative for malaria test. Well, there are several reasons. One of them could be the diagnostic tool that you're using. But for my point of view, as an immunologist, I think it's also possible to have all the clinical symptoms, because what causes the clinical symptoms, like I said, it's not the parasite, it's not the parasitemia. And the diagnostic tool that we use most of the time is to look at the parasitemia, to look at the parasites on the slide. And so someone could have a parasite, very few parasites that are not detectable on the slide, but still get lots of clinical symptoms, because the clinical symptoms depend on how strong you respond to the parasite. So some people, those who are prone to severe disease, they tend to excessively respond to few parasites, whereas those with asymptomatic infections, they can have um, huge amount of parasite, a huge parasitemia, but no symptoms at all. That's because they're not responding. Their immune responses are not strong. They're very, very modulated. They are very, very regulated compared to those with a severe disease who excessively respond to very few of these parasites that are not even detectable on the slide. But it could also be that someone has been taking medication, or there's a other practical aspect of not detecting malaria when someone has clinical symptoms. But at an immunological point of view, it's very possible, because the clinical symptoms are due to the immune responses to the parasites.
Oh,
Thank you,
thank you
too.
very much. I hope the questions are over.
Yeah, yeah. Thank you very much for your time and for your interest. I can share. Rashid, you can also share the
Thank,
well, well, I'll have to ask first before you share the slides to the public, because I've put some data from one of our PhD students which are not yet published. So I will first ask him the permission before you share it with everyone. Otherwise, I don't mind sharing the slides.
thank you very much, Prof. We cannot thank you enough for the wonderful job you've done. And I must say that we've learned a lot from your presentation. And also to the organizers, thank you very much for putting this thing together. And also to all the participants, thank you very much for making it a date with us. I believe that whatever we've learned from the Gospel of Professor, we'll take it to our local scientific communities and then share the knowledge with people. As we conclude, let us remember that science knows no country because knowledge belongs to humanity. Thank you very much. I hope, Prof, next time we call on you, you will open your two arms to attend to us and accept our invitation once again.
Yeah. Okay. Thank you very much.
Thank you very much. Bye bye.
Bye bye.